the University of Illinois in plant biology and crop sciences, and I'm also a professor at the University of Lancaster in the UK. The 50s and early 60s saw large-scale famines, some due to conflict and poor policies, but others because the world simply could not produce enough food to support its growing population and demand. The burden falling, as always, on the poorest. The Green Revolution provided the means to grow sufficient food and was a major contributor to ensuring supply could meet demand for the next few decades. However, the technologies of the Green Revolution are meeting their biological limits, and as reported by the UN FAO, the last 6 years have seen an accelerating increase in those who are calorie insufficient. And again, this is not just in conflict zones. Particularly affected are countries of sub-Saharan Africa and the poorer countries of Southeast Asia. Ironically, these are the countries forecast to see the greatest population growth. The Green Revolution was founded primarily on genetic improvement of our major food crops. That was breeding our major fruit crops so that more of their biomass was partitioned into the parts of the crop we eat. For example, the grains of our major cereals. Much of this was achieved by dwarfing, so shorter stems and more grain. The genetic innovations were accompanied by agronomic innovations to support these new revolutionary varieties. One of the most effective actions for helping the poorest, often smallholder farmers, is to provide better seed, which the Green Revolution did. Better seed can transfer from farmer to farmer. Before the Green Revolution, wheat, rice, and maize cultivars had a harvest index of about 30%. That is to say, 30% of their shoot biomass was grain. By the turn of this century, the best rice and wheat cultivars were achieving harvest indices of 65%, and maize and soybean 55%. If there is to be some stem and structure to support grain at harvest, it is hard to see how further improvement in harvest index could be achieved, and indeed, the rate of improvement in crop yield potential is slowing, largely as a result of this. In his 1997 address to the Royal Society, the eminent wheat breeder and physiologist, Lloyd Evans, looked at the prospect of achieving the need for a doubling of food supply by the middle of this century. To quote from his article to the Royal Society, "It is not apparent how a doubling of yield potential can be achieved unless crop photosynthesis can be substantially enhanced by genetic engineering. Photosynthesis would appear on the surface an obvious target. It is, after all, directly or indirectly, the source of all of our food. Further, its efficiency, even in our best elite cultivars, is less than 1/3 of theoretical, so we are a long way from its biological limits. Yet, the photosynthetic efficiency of our best cultivars today is little different from that of their wild relatives and pre-Green Revolution cultivars. Why so much progress with harvest index and not photosynthesis? Improving harvest index was often the case of knockouts by mutation of just 1 or 2 genes. Photosynthesis involves over 100 genes, and most knockouts will be lethal. So why is there now a chance to improve photosynthesis? First, it is the best known of all plant processes. We know the underlying genes, the structure and function of the products, the metabolic network, and its interactions with the environment. And we have high throughput technologies to measure photosynthetic efficiency. Secondly, this knowledge allows us to simulate the whole process using high performance computing, treating the process like a production line and predicting where to distribute resource, e.g. nitrogen, to obtain the maximum efficiency. Such approaches have identified a number of bottlenecks that could be solved by upregulating specific genes or introducing alternative metabolic pathways. Using tobacco as a model crop, that is the RIPE project, has now demonstrated and published 3 very different innovations that have increased biomass production, each over 20%, in replicated field trials in Illinois. RIPE is a multi-center international project funded by the Gates Foundation, the Foundation for Food and Agricultural Research, and the UK Foreign, Commonwealth and Development Office. Tobacco was used because it is easy to transform and produces large amounts of seed, allowing field testing within a year of transformation. So it is essentially a model telling us, is the prediction one worth taking to food crops? Doing this in a food crop would take about 3 years. However, there has been considerable skepticism that this could be translated into a food crop. But last summer, we showed a similar, more than 20% increase in yield with the first of these technologies in soybean. Most importantly, this increase in yield was achieved without any loss of seed quality, suggesting that increased photosynthesis not only provided more resource for seed production, but also for the end-fixing brachyrhizobia, since the crop, as per standard practice with soybean cultivation, was not nitrogen fertilized. This, though, I would argue, is the tip of the iceberg of what is possible in manipulating photosynthesis. RIPE has shown now it can be done, but much more than RIPE is needed to use photosynthesis to provide sustainable food security. Looking at all inefficiencies in crop photosynthesis suggests that with a concerted effort, and well beyond the RIPE project, a doubling of crop photosynthesis, and in turn, crop yield could be achieved. And it could be achieved sustainably, because increasing photosynthetic efficiency often doesn't require more water and not necessarily more nitrogen. Imagine we could be providing the poorest farmers, and indeed all farmers, with seed that could double their yield, benefiting the environment by avoiding the encroachment of agriculture onto yet more forest, avoiding the need for more irrigation water, and combined with end-fixing technologies, avoiding the need for more fertilizer. Achieving, though, that requires a concerted, multidisciplinary international effort, from improved genetic engineering through to seed distribution systems, and of course gaining social acceptability, especially in the countries and with the smallholders most in need of greater and sustainable food security, ideally done as an open science project. Thank you for your attention.